the research lab or the unit for the Algae, so that our student or faculty member, they also work in this case. And this is also very new to me, because most of the professional pharmacists and we are the working in the related to the pharmaceuticals, like a chemistry, analysis, ecology, regulatory affairs, and clinical research, pharmacy practice. Most of time we are the involved in the testing as well as the dosage form design, including the stability. Nowadays we are the using the number of the software which is related to the dosage form, either it may be CAD, QVD, and nowadays also we are the working on the pharmacodynamic. So I hope that this field we also want to the include in our institute, and today's webinar is very very new to the each and every person. So from core of my heart, I welcome you, and proceed please, Dr. Amanji. Mute, Amanji, please unmute.
Okay. Thank you so much, sir, for your words. So before proceeding far further, I want to just introduce Dr. Ambika. She is algae expert and founder of Proalgaetech. She is expert in algae cultivation techniques and harvesting methods. Currently, she is involved in algae ingredients substitute for the food industry's food supplements from microalgae. She has studied about subaerial samples of collections to microscopic identification and also systemic taxonomy of the microalgae. During the after the research, she has published 6 research papers on microalgae with peer-reviewed journals. So she is now algal culture technical supporter. She is working as advisor speaker in Germany. So can you proceed, ma'am?
Yes, yes, Amanji. So thank you so much, Dr. Aman and also Dr. Gupta, for inviting me for this webinar. It's an honor to talk about algae and to share my experience and my knowledge with you all. Shall we start?
Yes, madam.
Yes, ma'am. Please go ahead, please.
Yes, I'm going to share my screen now. Could you please allow me to share the screen, Dr. Aman?
Yes, ma'am.
So can you see my screen now? India. I've been awarded doctorate in 2018. Since then, I started my own business in Germany. I'm the founder of Proalgaetech. So Proalgaetech is basically algae consultation and technical support company. We are working from small scale industries to large scale algae industries. We provide a technical support from algal strain selections to cultivation and also algal harvesting techniques. We are also supporting and for the algae ingredients for food and pharmaceutical industries. So today I'm going to cover these following topics. So what are the definition and the brief classification of algae and the techniques which is involved in the cultivation system of microalgae, bioactive compounds from microalgae and their types, what are the physiological effects of algal bioactive compounds and what are their uses, algal drugs, algal uses in pharmaceutical industry, and I'll be concluding with the remarks and future perspectives. So let's have a brief note on these algae. Algae are very simple photosynthetic organisms that have wide range of applications. This group of phylogenetic algae, it's having more than 30,000 species in general, but these can be categorized as multicellular microalgae and unicellular microalgae. So here in the left side, you can see Chlorella vulgaris, which is a microalgae, which is unicellular microscopic algae. In the right side, you can see Caularpa racemosa, which is macroalgae, which is multicellular, and we also call it as a seaweeds. So we all know that. So the photosynthesis is a process by which light energy is converted to chemical energy, whereby carbon dioxide and water is utilized and oxygen is released, and which is converted into organic molecules. And these organic or chemical compounds with a particular biological activities, we call it as bioactive compounds. So this process happens in microalgae just like plants. As like plants also depends on sunlight and carbon dioxide for the photosynthesis. It releases oxygen with the bioactive compounds. Likewise, algae also does the same thing. It also utilizes the carbon dioxide as a carbon source, water, and it absorbs sunlight as an energy source, and it releases more than 10 times more than oxygen than these plants. So with these biomasses, we have multiple potential we are going to discover in the upcoming slides. So I'm going to give you the brief classification, not in detail. Algae classification is microalgae, as I mentioned, microscopic algae, which comes under the kingdom Protista, which is having 4 phylums: Cyanophyta, Pyrophyta, Chrysophyta, and Chlorophyta. Cyanophyta, cyano is nothing but blue-green algae, which is, for example, nostoc and anabina, which show in the figure in the left side first and second, which is nostoc and anabina respectively. Pyrophyta, dinoflagellates. Chrysophyta is a diatoms or golden brown algae, we call it as, for example, Pinularia and Chrysophyta species. The middle two pictures shows that Pinularia and Chrysophyta. Chlorophyta is nothing but chloro is green algae, which is microscopic green algae, for example, Hematococcus and Chlorella. The last two pictures shows the Hematococcus and Chlorella species respectively. Another part of classification is microalgae, that is seaweed, which is also comes under Protista, which is having 3 phylums: Chlorophyta, Rhodophyta, and Phaeophyta. As on the basis of the pigments of the color, it has been classified. Chloro is nothing but green algae. Rhodo is nothing but red algae. Phaeophyta is nothing but brown algae. For green algae, it is Gracilaria cordium, which is green color. These are 2 examples. Rhodophyta red algae, Russell and red razor, which is in the left side pictures. These 2 are red algae. Phaeophyta, it's a brown algae. Britannica species is one of the example for brown algae. So let's discuss in detail about the culture techniques for microalgae. So when we talk about culturals, we have several techniques are available for microalgae cultivation. The choice of the which technique is to be chosen is mainly based on the selected microalgal strain as well as its application. Why we should choose a right strain? Choosing the right strain for production is an important decision that can have significant impact on yield and quality of our final product. When choosing the strain, there are several factors to be considered. For example, growth rate. Choose the strain that has fast growth rate and can efficiently use light, nutrients, and carbon dioxide to produce biomass. It's a very important. How it can grow fastly or slowly according to the local regional temperature where you wanted to produce these microalgae. Next is adaptability. We have to choose the strain that can grow well in specific conditions of the open pond or PBR method that you will come to know in the following slides, which includes temperature, pH, light, and nutrients. How these strains are very much adapted to these physical factors like temperature, pH, light, and nutrients, because these factors is completely dependent on the strains, how it will be adopted, how well it is adopted, and so that if it is well adopted, you can expect a well biomass and well yield out of this. And another important factor is that nutritional profile. We have to choose a strain which is having high protein content. For example, chlorella and spirulina, which is having 30 to 40% nutrient or protein content, and which is having very well balanced amino acid profile, and also which is having high level of important nutrients such as vitamins, minerals, and pigments. Another important factor is that stability and consistency. We have to choose the strain that has a stable and consistent growth and production pattern with minimal variation from batch to batch. So when you initially, before we go for pilot scale, we do inoculate, we do study the consistency and stability of the strains. It has to be maintained. The consistency has to be maintained batch to batch. If more variations you could notice from batch to batch, then you should not consider that strain to be grown in the future. Why? Because when you choose not stable and consistent growth in the production pattern, then you cannot expect a very good yield out of these trees. So these are the factors we should be considered when we are choosing the strain. So that is the reason the choice of technique is mainly based on the selected of microalgal strain. Suppose the strain is very sensitive to physical factors or very sensitive to other organisms, I mean to say contamination, then we should not go for open pond system of cultivation. We should cultivate them in closed culture system. Here in a schematic representation, I've shown that algal culture techniques has been divided, has been like following in two system, different system type of cultivation. One is closed culture system, another one is open culture system. As I mentioned, open culture system may is a cheaper technique, but the thing is, all strains will not be suitable for open culture system. So when you get to know that, when you considering the following, the factors which I shared with you, so that is very sensitive to open pond culture system, then for that strain you should follow the closed culture system. Okay, the strain is very stable. It has a consistency. It has quite a well adaptability nature. Then you can go for open culture system. Open culture system and closed system. These are main two system of cultivation technique, culture technique. So open culture system is quite less expensive. We can call it as a cheaper investment, but the contamination is very high here in this case, open culture system. As the name shows, open culture system, it is we are cultivating them openly. There is a more chances of getting contamination. That is the thing. When you compare to closed culture system, it is quite expensive. And but the thing is, you can cultivate the culture in a controlled manner, so there is less contamination you could expect from this from closed culture system. It's completely depend on the strain which you have selected. So choice of culture media, choice of cultivation system, maintenance of this abiotic and biotic cultivation parameters are the main aspects of microalgae cultivation. So the most common cultural techniques used in cultivation is photoautotrophic culture. As the name indicates, it is light dependent culture. So this method involves growing microalgae in a controlled environment using light as an energy source and carbon dioxide as a carbon source. Another is heterotrophic culture. This method involves growing microalgae in nutrient-rich environment using organic carbon source such as glucose or yeast extract as a source of energy. So next method is open pond culture. In the first two pictures, you could say circular pond type of open pond system and raceway ponds. Here we should consider when you select the open pond system, you should also consider a few points here. So the size of the pond, it is also a vary. It is depend on the production scale and also a desired yield. So for the commercial production, open ponds are range in size from several thousand square meters to several hectares. The size of the pond is usually depends on the factors as daily production target, the area required for mixing and pumping system, and also the surface area need to be provide sufficient light for photosynthesis. Here, one thing we should consider the depth of the open pond should not be more than 30 centimeters. Why? Because the sufficient light penetration it won't be possible if it is more than 30 centimeter in deep. Photosynthesis plays very important role in algae, just like plants. Here, the sunlight plays very important, crucial role in photosynthesis. When there is no sufficient light is being provided, then you cannot expect any further very good quality yield of biomass. So that is the reason. In small scale, for example, open pond can be much smaller, ranging from few square meters to few tens of square meters. So the size of the pond can be adjusted based on the amount of yield, amount of the produce, how much you wanted to grow. That is also depends. So here in open pond system, as I mentioned, the maintaining of physical factors like temperature, pH, water salinity, nutrients availability, light penetration. So it is very important. So in open pond system, the mixing of the culture, either in open pond or photobioreactor type, the mixing of culture is very, very important because when the cells are mixed properly, the photosynthesis and metabolism happens uniformly. So that is very important. So that is the reason the mixing and the light penetration, the supply of carbon dioxide, the peddling system of open pond system should be very proper and it plays very important role, and it is completely, it's directly proportional on the yield of the growth of any selected strain. Next method is photobioreactors. These are closed system designed for controlled microalgae cultivation and used to produce high value microalgae products. What are these high value microalgae products? So suppose in your field only, pharmaceutical products, pharmaceutical ingredients should have high quality, should maintain high value quality points. Your whatever the ingredients you're getting it out of these microalgae should be maintained abiotic environment, should be out of contamination. The quality of the biomass should be very well to consider in the pharmaceutical industries. So that is the reason photobioreactors type of cultivation followed when you are expecting your when you're expecting your biomass is going to be used in food industry or pharmaceutical industry, then you have to go for closed system type of cultivation just to maintain the well quality and also the controlled environment for the biomass. So as I mentioned, so photobioreactor is a type of closed controlled environment. It's a provide, excuse me. These photobioreactors has to provide a necessary light, temperature, and carbon dioxide levels for for microalgae cultivation. And it also eliminates the contamination from other microorganisms and pollutants. So these photobioreactors made from various materials such as plastic bags, tubes, or columns and come in different sizes and designs. So what there is an advantage of using this photobioreactor type of cultivation because it improves the productivity, it improves the product quality, it reduces the cost compared to traditional open pond cultivation. However, it is also required specialized equipment, technical knowledge, and greater operational costs. So operational costs are definitely very high, but when you are targeting a very good quality of microalgae product, then you must go for photobioreactor type of cultivation. Another method is fermentation. This method involves uh, we we grow the microalgae in a bioreactors under anaerobic conditions, which are suitable for certain species of microalgae. So, in conclusion, the choice of cultural technology for microalgae cultivation will depend on the specific requirement of the project, including type of microalgae being grown and desired end product and the scale of the production. So factors, which are the factors will be limiting for this growth? So the main factors will be temperature, salinity, light intensity, photoperiod, and pH. So the temperature range will be 16 to 27. Optimum temperature is 18 to 24. Salinity is 12 to 40. And optimum is 20 to 24. Light intensity is 10,000 to 10,000 lux and 2,500 to 5,000 lux. The photo period light dark. Light will be 16 hours. Minimum of dark is 8. For few strains, complete light phase is required. So this light is very important. Photo period factor is very important here. In recent research, it has been showed also not only a complete light phase, or you can call it as a photo period phase, is important because it has been proved that by exposing the culture in more dark reactions have shown a more yield also. But being said that, this is also completely depend on the strain, on the species, because few species are very light sensitive, few species are very sensitive to dark. It's so that is the reason we need to be considered, we need to be studied which strain has selected and with how much light period it is dependent and not dependent. So this is very important. The I would say the 80% of the yield it depends on the light. The culture, if you want to get a very good culture yield, then you have to maintain the light and dark phase properly as according to your strain selection. Then pH is also very important. It has to be optimal 8.2 to 8.7, and definitely each species has its own pH level. So microalgae harvesting and drying techniques. We have chemical methods, biological methods, and physical methods of techniques of harvesting. The choice of best method for harvest microalgae, it depends on several factors, which is including type of culture and use of microalgae, availability of resources. But most commonly used methods are centrifugation and flocculation. This centrifugation is high-speed centrifugation to separate the microalgae from growth media. It is suitable for large-scale production and it is widely used in commercial spirulina and chlorella production. So flocculation, this method uses chemicals or physical treatments to cause the microalgae to form flocks, which can then be separated from growth medium to sedimentation or filtration. It is relatively low cost and simple, but can also result in losses of microalgal quality. So first we'll take algae suspension and we'll do the screening, we'll do the thickening, And in the thickening, we can go for coagulation flocculation method, bioflocculation, gravitational sedimentation, flotation, and electrical based methods. Then you have to dewatering, after dewatering, filter it, centrifugation is then drying, and the biomass process of you can use it into biofuels or pharmaceutical and other aspects. So biomass applications. So why I've been showing in this slide only a 6 species. So these 6 species are most cultivated species in Europe. I'm talking about. So, for example, in Chlorella species, this species is produced in 11 European countries, and this species has high protein up to 60% and carbohydrate content, and it is also rich in vitamin B. So this chlorophyll is considered most of the species this chlorella species is considered as one of the most species grown in European countries. And when it comes to a protein, it is you can say that 60% of protein which is present in chlorella species. So this protein has been using in food industries as a food ingredient and also in pharmaceutical industry. When it is comes to nanochloropsis species, it is an important source of EPA and biofuel. So now nanochloropsis has been studied and being also studying for the biofuel alternative energy source because it is having 60 to 70% lipid content in it. So it is a very important source for EPA and also for biofuel. Hematococcus pulvalis. Hematococcus pulvalis it has the capacity under the stressful conditions to accumulate high quality of bioactive compound called astaxanthin, which is high value antioxidant carotenoid. Indeed, it is a leading natural source of commercial astaxanthin. So this one of the bioactive compound which has been extracted from Hematococcus pulvalis has it's been using in commercially, and it is available everywhere commercially because the astaxanthin, it is the high value antioxidant carotenoid which has been using in pharmaceutical industry, food industry, fish feed industry, and etc. So that is the reason Hematococcus pulvalis also plays a very important role in biomass applications. And Dunaliella salina. Dunaliella salina is attracted by by different industries because of their antioxidant beta carotene source. It is an excellent natural source for beta carotene, antioxidant beta carotene, and it is also available commercially nowadays. Spirulina, next to chlorella, you can consider this is another species which has been grown which has been selected from different industries. It is a very rich in protein and antioxidant compounds, and it is also used in the extraction of pigments such as phycocyanin. Phycocyanin is one of the bioactive compounds which you can get it, which is in blue in color, which is used in health, cosmetics, and food applications. Now, Proalgaetech is also having both this phycocyanin, astaxanthin product, and we are supporting food industries, pharmaceutical industries, feed industry, and we are also supplying tons together this phycocyanin and astaxanthin for their commercial use. Porphyridium species. Porphyridium species is used for the extraction of exopolysaccharides and pigments. We can call it as phycoerythrin. So these species also produce compounds associated with potentially beneficial health bioactive, for example, antifungal, antiviral, anti-carcinogenic, anti-inflammatory, antioxidant, and immune system stimulant. Thus, they are used in variety of health products, including tablets and capsules. So, not only a food supplement or nutraceutical, but also in cosmetics and feed industry, these applications have been diversified because, for example, astaxanthin and phycocyanin it's having so well demand in cosmetics as for the natural colors in the cosmetic industry, and also astaxanthin plays very important role in shrimp's feed because shrimp's feed it gives a natural orange color to the shrimps and equal uniformity growth you can see in the shrimps also after you started giving this astaxanthin as a feed. So not only in a food supplement, because before this biomass application was restricted only to the food supplement. So when we heard about this microalgae, we used to think that okay, this is a food supplement. So when we take this spirulina and chlorella protein supplement, it will balance our protein in our body. But it is not restricted to only particular industry. So not only in nutraceutical but also in cosmetics and feed, these biomass applications is very well diversified. So use of algae formulations with potential applications in new researches. As I mentioned, it is not only restricted as a food supplement, it has been diversified as a natural colorant, animal aquaculture, poultry feed, renewable energy, wastewater treatment, as a fertilizer, pharmaceutical and nutraceutical, and also cosmetics. In this schematic representation, you could see so this microalgae is being placed in a center. It has covered a 360 degree of its potential in almost all the industries, from food industry to cosmetics. It has been used as a natural colorant. For example, phycocyanin, astaxanthin. Nowadays, it's been booming in food industry, and it is using as a natural colorant a blue and orange color from spirulina, from spirulina and Hematococcus, and animal and aquaculture poultry feed. It has improved the animal and aquaculture growth and also fish color and meat quality as well. So here being said that fish color and meat quality, which is helping to increase the quality of the food or animals' health. So, and one more point is in plant-based meats also they are coming forward to use this microalgae-based ingredients in their plant-based products. For example, plant-based meat for the natural colors, which gives a red color for the meat and fish color, salmon color for the fish from Hematococcus pulvalis, astaxanthin. When it comes to renewable energy, from the past 20, 30 decades, it has been researched on this microalgae about alternative energy sources such as biodiesel, bioethanol, biogas. But the thing is, because of underdeveloped technical cultivation, we are not yet successfully succeeded in this renewable energy. But we are the research is still going on this by developing the technicals by overcoming of these technical challenges. Definitely algae plays very important role in an alternative energy source. And wastewater treatment, we all been what neglected when seeing the algal growth in the ponds. We think that okay, they are the poisonous. Yes, they are the poisonous, but it happens with the eutrophication. It's a man-made pollutant. But we use microalgae also to remove these nutrients such as nitrogen and phosphorus to prevent eutrophication and water pollution. Because the nitrogen, phosphorus, what we consider as a poisonous for the pond, for the water, then it is a nutrient for the microalgae. So that is the purpose we use in wastewater treatment also this microalgae. This microalgae will be taking up this nitrogen and phosphorus to prevent the eutrophication and water pollution from the water bodies. And fertilizer, as a promising alternative to chemical and synthetic fertilizers, now we have already available the commercially seaweed-based fertilizers more than microalgae-based. But microalgae-based biostimulants is also available in the agriculture field. So not only our wastewater, not only our renewable energy, it also plays very important role in the fertilizers. When it is comes to pharmaceutical and nutraceutical, due to their high bioactive properties, they have potential for the development of pharmaceutical and nutraceutical. We'll study in details in upcoming slides about pharmaceutical and nutraceuticals. When it comes to cosmetics, due to their bioactive content, microalgae products have been used in the cosmetic industries as an antioxidant, sunscreens, and moisturizing agents. So, in overall, we consider this potential. It is 360-degree potential. You could also see There's algae formulations, which is having from food industries to cosmetics industries. So in food supplement, we consider spirulina and chlorella is the most commercially used species in human nutrition because it's having high protein content and excellent nutritive values. So bioactive compounds. So basically, what are bioactive compounds? So these bioactive compounds are naturally occurring chemical substances that is having biological effect on living organisms. They found in plants, they found in animals, they found in microorganisms, and they are often responsible for therapeutical or medical properties of many foods and natural products. These bioactive compounds include wide variety of molecules such as vitamins, minerals, antioxidants, flavonoids, polyphenols, terpenoids, alkaloids, and many others. They can also act on human body in different ways, such as modulating cell signaling pathways, reducing the inflammation, and preventing the development of chronic disease, and so on. Does it in the extensive research? It's also increased in such a different fields such as nutrition, pharmacology, biotechnology, and as they hold great potential for improving human health and well-being. So, why microalgae based bioactive compounds? Because microalgae has fast growing and solar powered biofactors when it's compared to other factors like plants, animals. So, there's microalgae is a fast growing and solar powered biofactories. It's an ideal platform that poses potential to meet the enormous demand of pharma, biopharmaceutical, and human nutraceutical production. So, algae, which are rich source of these bioactive compounds, including polysaccharides, polyphenols, pigments, and fatty acids. Some common bioactive compounds found in algae are carotenoids, phycobilin proteins, fatty acids, polysaccharides, and polyphenols. So these compounds have been studied for their potential health benefits, including antioxidant, anti-inflammatory, and anti-cancer properties. So, what are the advantages being selecting microalgae based bioactive compounds? Higher production yields of bioactive compounds through solar energy and carbon dioxide utilization, shorter growth period with doubling time, less than 24 hours, especially for large scale production, no competition for our need to occupy land areas. When you select plant based, definitely we have to compete with the food industry, with the land utilization, but here no competition. Relatively simple supplementation of climate condition and controlled bioactive compound production by regulating growth conditions. So these are the advantages when you choose microalgae based bioactive compounds. So, how they obtained? Bioactive compounds from algae can be obtained directly from primary metabolisms such as therapeutical proteins, fatty acids, vitamins, and pigments, or can also be synthesized from secondary metabolisms such as compounds can present antivirals, anti-algal, antifungals, anti-enzymatic, and antibiotic actions. So many of these compounds, for example, cyanovirin, linolenic acid, oleic acid, palmitoleic acid, beta carotene, vitamin E, B12, beta carotene, phycocyanin, zeaxanthin, lutein have antioxidant, antimicrobial, anti-inflammatory properties with the potential for the reduction and prevention of diseases. So these are the examples of these bioactive compounds. So phycocyanin, you can see, and DHA, EPA, lutein, zeaxanthin structures. So what are the physiological effects of these algal bioactive compounds? So PUFAs are the most studied microalgal lipid compounds. These renewable bioactive lipids have been used in prevention and treatment of cardiovascular diseases. So a previous study showed that this dietary omega-3 PUFAs have protective effect against these heart diseases and derivatives of PFAS, namely DHA, EPA, alpha linolenic acid, and DPA, have been applied to treat type 2 diabetes and also inflammatory bowel disorders, skin disorders, and asthma. So DHA EPA have been demonstrated to reduce medical complications associated with strokes, arthritis, and reduce hypertension while acting as anti-inflammatory agents. What are the advantages? Because we have in the market, like other than synthetic, you can call it the synthetic bioactive compounds or plant-based or animal-based, usually animal based micro and the other microorganisms based bioactive compounds. So what how these microalgae based bioactive compounds stands out? So microalgae offers the following advantage. I'm comparing here with the fish oil. So one is the proof of profile is more straightforward. So what is mean by straightforward here? It's the most effective than the fish oil. So cultivation condition can be controlled here in a microalgae, as we discussed, either in open pond or in the closed pond. So definitely for pharmaceutical ingredients, nowadays we go for the closed type of system cultivation, and there we cultivate this microalgae with well environmental controlled conditions. This shows that the quality of the PUFA profile is very well than fish oil. So different algal species produce different PUFA. This is one advantage here when you compare to fish oil. So fatty acid production from microalgae has a potential to be less expensive than fish. When you compare to fish, PUFA profile from the fish, it is well less expensive than fish. So these microalgal fatty acids have no odor, metal contamination, or cholesterol. When you talk about fish oil, definitely the fish oil profiles completely depend on the feed, what you will feed for the fishes, and the water where you either in the pond, however you are cultivating the what you are growing the fish, that is also depends. The metal contamination, the cholesterol, the odor of the fish, it is completely absent. You cannot expect from this microalgal fatty acids. It has no odor, no metal contaminations, and no cholesterol. So the EPA esters obtained from microalgae are more stable than the fish algae products, and also the lipid levels usually 20 to 50%, but can accumulate up to 80%. So with the genetical engineering or with the playing around the environmental condition, we can also increase the lipid levels from 20 to 55% to 80%. This we cannot expect from the fish oil. So we cannot alternate the lipid content or the lipid levels which is present in the feed. Rather, in the microalgae, we can accumulate up to 80% by altering the environmental technical factors, and also by using genetically engineering species or strains. So under stressful conditions such as nitrogen starvation or elevated temperature, see, so you can up to 80% accumulate, made the microalgal strains to increase its lipid level by placing the strains under the stress condition. For example, nitrogen starve. If you stop supplying this nitrogen, then it will increases the lipid level. If you increases the temperature, it increases the lipid level. So this can be altered or as per our requirement, but you cannot do it with the fish. So here I'm giving you which are the main species which is involving in this bioactive compounds. This is one of the diatom called Phaeodactylium trichonutum. So this is a diatom. It is the only species in the genus Phaeodactylum. Unlike other diatoms, it can exist in different morphotypes: fusiform, terradiata, or oval form. It changes in cell shapes, can be stimulated by environmental conditions, which is also known in connection with EPA, which is rich in EPA. Another species is Schizochytrium species. So I have been worked on this species. We have cultivated in heterotrophically with the fermenters by using sugar carbohydrate as a carbon source. And we have right now this DHA EPA product with us, and we are using this DHA EPA powder for the fish feed, and we have got very well results from these products. Schizochytrium is very rich in omega-3 fatty acids, DHA and EPA, and therefore often used for the production of algae oil. Algae oil also we have been extracted from this schizochytrium species. So, I would like to give a brief on this polysaccharides also. Because of time shortage, I have taken only three bioactive compounds. One is PUFA, another one is polysaccharides. So, polysaccharides are biopolymers in which less than 10 monosaccharides are linked and linear or branched chains via glycoside bonds. So there are growing number of concerns regarding polysaccharides as high value component in pharmaceutical, food, cosmetics, fabric, and emulsifier applications. We all know that. So sulfated polysaccharides from microalgae contain sulfate esters and possess attractive pharmaceutical application including antioxidant, anticoagulant, anti-inflammatory, antiviral, antibacterial, anti-tumor, immunomodulatory, and radioprotective properties. So, sulfated polysaccharide from cyanobacteria, spirulina, have been used as antiviral drugs both in vivo and in vitro. While polysaccharides from eukaryotic microalgae such as Chlorella vulgaris, Cinedesmus, and Dunaliella species have high inhibitory viral activities. And on these species, still research is also going on the further for the COVID anti-COVID viral drugs, but it has not yet announced that they have got a results and so on. But research is happening on this species. So this is Chlorella vulgaris. It's a green algae. Um, Polysaccharides have high immunomodulatory activity, though therefore extracellular polymeric substances containing polysaccharides from Chlorella vulgaris are the promising agent for chronic airway inflammation prevention. The primary pathogen mechanism of many respiratory diseases, including bronchial asthma. So another is Emetococcus pulvalis, which is having polysaccharide fraction HPPC3S1 obtained from Hematococcus pulvalis. It is a paranoid containing amino acids O acetyl groups and shows substantial immunostimulatory effects on splenocytes and B lymphocytes. So, what are the pharmaceutical uses of algae and algal drugs? So, microalgae contain high valuable bioproducts used in cosmetics, food supplements, some drugs used in treatments from different diseases. Whereas microalgae play a big role in developing anti-cancer drugs, a huge volume of research and bioactive compounds from well-studied algal forms such as Arthospira, that spirulina, Chlorella, Dunaliella, nostoc, led to identification of antimicrobial, antiviral, anticoagulant, anti-enzymatic, antioxidant, antifungal, anti-inflammatory, and anti-cancer activity. These studies have based on extraction of bioactive compounds, and these microalgae having each algal species having different uses and different bioactive compounds. For example, Chlorella we use as food supplement, formation and regeneration of blood cells, anticoagulant, anti-tumor, antioxidant, and antibacterial effects. Dunaliella, antioxidant, bronchitary, and hypertensive, muscle relaxant. So for all these activity, you use it as antibiotic substances also, which is having antibiotic substances. When it comes to nostoc, we used as dietary supplementary treatment for fistula, anti-inflammatory, and anti-cancer. Spirulina, we call it as a superfood used in food industry, medicine, health, benefits property like antioxidant, anti-cancer, antiviral, antibacterial activity. Have positive effects against obesity also, diabetes, malnutrition, hyperlipidemia, anti-inflammatory, and anemia. So these are the pharmaceutical uses of few species. I have taken only a four species. So what is the how to like overcome, how to compete with the locally available commercially with the different system of production and therapeutical proteins. So not only microalgae, so other bacteria, yeast, plant based, insect based, human cell line based bioactive compounds, which is already proteins which is present in the market, how to compete with this and how these microalgae stands out, and how we can compete in this. It is not so easy, but yes, it is competing slow. It is competing with other uh, other different systems also. For example, I've taken a few properties here just to give you the comparison with the other uh, origin of bioactive compounds or proteins. So production cost when it's compared to bacteria, yeast, plant, insect, and human cell, algae is quite inexpensive. And human pathogen, definitely it is non-susceptible. When it is insect is susceptible, plant and yeast is also non-susceptible. When it is compared to contamination, bacteria, definitely bacteria, yeast, and human cell line comes under high risk of contamination. But in algae, it is very low. But in case of we are using photobioreactor closed system type of cultivation, definitely the contamination level is very, very low when it is compared to others. So glycosylation, it's also non-glycolization we can expect in this, other than human cell line and yeast, which is a glycosylation. Therapeutically efficiency is very, very high when you compare to other bacteria, yeast, insect, and humulene. Plant and algae are placed very, very high therapeutical efficacy. So half life is very high, and production time. This is very important. Production type. Bacteria, it's okay, it's a few weeks to month you can get it. And plants is also falls, no, it won't falls in the weeks, it definitely takes a years together. And animal cell also. And when it is comes to algae, within a month, from weeks to month, you can get production very easily. So the current market, it's depend on 32% we are depending on bacterial based, 15% we are depending on yeast, less than 10% is plant, less than 10% in English insect, and 43% we are dependent on human cell line, and less than 10% in algae. Why? Because algae is now getting into spreading its business strategies into different fields. Now it has been started. So definitely in couple of years, it will be more than 40%. So what are the economical potential of this algae? As we discussed in previous slides, as I mentioned, it's placed in a food chain, it's placed in a feed industry, sewage treatment, fish culture, and also it's placed in plays a role in alternative energy sources also. The thing is, yes, it has an economical potential of algae, but what are the challenges to explore this potential of algae? So the first basic thing of the challenge in a technical is we are still following underdeveloped technical technology for producing this microalgae. So, and research is happening, it's going on how to overcome these challenges, because the cost of cultivation, it's very high comparatively, and the harvesting cost, the 30 to 40% of the cultivation cost is going for harvesting. So we need to work more on this culturing. So being having this technological challenges, so definitely the economical potential, it's not diversified so well, it's somewhere lagging behind because of the cost of the product. Definitely when we are getting a very easy comfortable synthetic products in the market, with a less price. Why a fisherman, why a food industry, why a feed industry want to have this microalgae based ingredients, though it is having um, so many what do you call, so many properties, good properties. The thing is the cost. We need to work more on this. Other than this, definitely economical potential of algae is very vast. So almost all industry, the food industry, feed industry, sewage, fish culture, all industries working on this microalgae. And definitely it's already spread over different industries. And it is having, I always say that 360 degree of economical potential you can expect from this algae. So conclusion. So microalgae have long been known as a promising platform due to their feasibility in modern manufacturing facilities, only using carbon dioxide, water, and sunlight to produce bioactive compounds. However, from the sustainability prospect, certain obstacles remains to be overcome for the use of this microalgae production system. Secondly, metabolites such as antioxidant, antiviral, antifungal, anti-tumor, anti-malarial, and other bioactive compounds have industrial potential. However, it remains a significant challenge that microalgae compete with current well-established platforms, partially due to a lack of secretion of recombinant proteins into the media. So this is very important. So having a product, it's not a big deal. How we compete with the well-established platform, with well-established companies, that is also very important. So that we need to work on the technology, we need to work on the recombinant proteins, recombinant strains, we can call it as. When we started using this, definitely we can be able to compete with the current well established platforms. So therefore the potential of microalgae as a cell factory has yet to be realized, because the awareness of this microalgae is still very less. So we need to work on these microalgae awareness, and the future effort should concentrate on exploiting their distinctive advantages such as their GRS status, inexpensive culturing, and possible scalability. Scalability, down process, up process, cost, culturing cost. We should work more on this to compete with well-established platforms. So bioactive metabolites of algal origin are a special interest in the development of new products for pharmaceutical, cosmetics, and food industries. Further research should be conducted with these bioactive compounds to verify their beneficial effects of for humans, their degradability when released into environment, and their effects when used in animals. So need to be consider all these factors. So these will be adopted to the ecological demands of strains and application aims for biomass, valuable substances, and ecology. So we are working on this bioactive compound called phycocyanin, which has been extracted from spirulina, blue-green algae, which is consumer demand for natural colors, best natural blue solution, vibrant blue, and nutraceutical applications. We are supplying this phycocyanin for food industries and also for the nutraceutical industries. And it is European market organically certified product, which is which is contaminated free, odor free, and also cholesterol free. And it is metal free also. It is recommended to use daily. So highly solubility for food, bakery, beverages application, and it is certified free from bacteria, microsystems, and pesticides. So composed of pure dried aqueous extract of the spirulina, suitable for vegan organic markets. Another product is astaxanthin. We are extracting this astaxanthin from Hematococcus pulvalis, which is having huge health benefits like eye health, bone joint health, brain, skin, and cardiovascular health. So it is also used as a dietary supplement. It reduces your inflammation, it boosts your immune system, it improves your cardiovascular health, skin, and it also protects you from the skin damage and so on. Thank you all for your patience to listening to my talk, and I'm very honored and glad to be part of this event. Thank you so much.
Okay, thank you so much, Dr. Ambika, ma'am, for your kind words. Ma'am has covered basic about the various types of algae with their physiological effects as well as the various bioactive compounds. It also covers the various conditions and temperature that are required or measured to grow proceedings. Ma'am, here are some queries that are raised by our students.
Yeah,
I'm happy to share that more than 150 participants are viewing you from our YouTube channel.
thank you.
My question is how these algae is different from probiotics.
How algae are different from, it's that source of organisms, it's just different because probiotics are getting from good bacteria, you are getting from good bacteria, and this is also considered as one of the source you can get it from the algae. So it is not completely differ from probiotics. The thing is the probiotics, it is which is, for example, I'll give you probiotics which helps for the gut health. If you take, it increases the good bacterial in your gut, for example. And when you take a probiotic based, algae based probiotic, which is having its own beneficials to your health.
Ma'am, during my PhD, I have used Saccharomyces boulardii as probiotic encapsulated with the drug for mesalamine for management of ulcerative colitis. And the things I have observed is the probiotics give synergism action to that drug moiety.
Okay.
Okay. Ma'am, one more question is there. Is there any market formulation that is algae based?
What?
Ma'am, is there any pharmaceutical preparation that is based on the algae?
Pharmaceutical preparation, we won't get like ready pharmaceutical preparations. It is completely it will work as a secret ingredient from each and every industries. Now we are just providing as a raw ingredient for different pharmaceutical and food industries. They do their own what do you call their receipt for their product. There is no particularly standard protocol, I would say, but for the fish feed or so, we have a particular dosage to be used, but not other. I won't say that there is a particular recipe or so for the pharmaceutical.
Okay, ma'am, one more question. If we talk about pharma products like your micro emulsions, your pellets, we always talk about their yield. So, what is the basic units in this case of algae? For example, if we talk about probiotic, they are available as CFU, colony forming unit. So, how can we can say that identify and what we can say that what is the basic unit for the algae? How we
You use
measure
it. We use it in milligrams.
milligram?
Mgs.
Okay.
Mgs, for example, for every day, for astaxanthin, I would say, daily diets as a diet supplement, you are not supposed to take more than 8 mgs. Up to 8 mgs are like advisable, not more than 8 mg. So we always consider in mgs, in
Okay.
milligrams.
Thank you so much. Now I would like to call our respected director, sir. Yes, Gupta, sir. Sir, please unmute कीजिए.
Hello, so Dr. Ambika ji, she has given the excellent information, and I know that this is not my field of the algae, but nowadays I hope that ke number of the opportunity in pharmaceutical sciences, but sometimes most of the people they are very afraid like this types of the activity because you know that if we working on the algae, number of the contamination may take place. So separation, and I have write down one word is the cultivation of microalgae. So this is the one of the technique itself, a technique, and if you want to the segregate for this purpose, NIDA obviously one of the expertise, but I hope that number of the research team person they are attached with this one, and we are running up the pharma cognosy department. So no doubt in the coming days it will be the also helpful for the treatment of the many chronic disease even that. But for this purpose need a one of the specialization separation maintain the you can say specialized condition and development and then purification convert in the form of the dosage form. So I hope that it will be the helpful to the researcher. And I also expect from the Dr. Amkaji, you can also give some of the guidelines how we can develop the algae in our department, because when the as a student life and algae just like a difficulty like a ponds and everywhere we can separate out and we can throw it without knowing that it's itself it have a, some of the medicinal value or pharmacotherapeutic importance. But nowadays people are the more and more worry about it. And madam, I want to know the any application of the algae in the cancer treatment like that.
Yes, sir. It has been showed few species has been recorded which is having anti-cancer also. For example, nostoc, but it research is still like practically show that okay, it is having it has been proved that it is having anti-cancer agent, but research is still going on. For example, nostoc, which is having anti-cancer agent, yes, it is. And you also mentioned that cultivating in the laboratories. Definitely, I have maintained I have recorded more than 100 cultures during my research. So the cultivation maintenance of culture is very important in the research. So for being so being in the pharmaceutical industry, so that is the reason you can start guiding your
Interesting.
students, your research scholars for taking up a few research on this, and definitely they will explore how we can maintain, how we can cultivate. For that purpose, definitely we can get it, we can outsource the ready-made media, the ready-made strains for the student studying purpose. So growing in laboratory case, pilot scale is different. We have different challenges, and when it is come to commercial, definitely we have different challenges. But for the students, definitely you can outsource the media. You can cultivate. You can go for antiviral, antibacterial, and also you can get it done. Antimicrobial activities you can get it done. Antifungal, antibacterial, all that you can get it done by suggesting your students to choose any one of the algal strain for their project report. So in this way we can differentiate working on this algae.
So thank you very much, madam. My only simple question: in which dosage form BA this is the more preferably given? Let us consider in which form, like a tablet, capsule, suspension. In which form it can be given to the patient, or you can say for the treatment of particular disease.
Which species you told, sir? I didn't. I just.
Any disease means I want to know that the algae in which format it is delivered or given. Let us consider it in a dry form. After that, it can be converted in which dosage form.
The use of these microalgae we consider as a raw materials. Use of the microalgae in pharmacy in different manners. For example, if you are using it as a supplement to improve the nutrition, either you can take it in a powder form or you can take it in
Yeah.
the capsule form. Suppose because I'm working with one of the research institutions in Germany, so they are just using anthocyanin as a bioactive compound. They are adding in their own tablets or their own pharmaceutical products. So it has different ways of using. So, for
Okay.
example, I mentioned as a supplement, as a food, as a diet supplement, you can take it in a tablet form and a powder form. But the thing is, the dosage you have to keep it in mind. For example, astaxanthin or spirulina or chlorella, we should not cross spirulina chlorella. We can take up to 6 mg per day. astaxanthin up to 8 mg per day. So the dosage should be maintained. And you that the product, I also inform you all that you are immediately, whichever it is available in the supermarket, you should check the quality. Should be maintained because as I mentioned, two differently we can cultivate like openly and with a controlled and a non-controlled. So that is very important. Right product is important and the dosage to be considered. So pharmaceutical ingredient having it's contacting us only for the ingredients as an ingredient. Phycocyanin, they order astaxanthin, or else they'll take it as take from uh, hematococcus, and they'll extract by their own. They'll give their own ingredients and they use in their perspectives. So it is completely different.
Okay, okay. So thank you very much, madam. So nice information, and I hope that it will be the helpful to all of us.
Yes,
Thank you.
definitely. We are also supporting different research institutions also in Tamil Nadu and Kerala. So definitely you can come forward and contact Proalgaetech for few for further research, for the collaborations, for international webinars. We will be conducting with the collaborating with different universities from Tamil Nadu and Kerala and and we introduce to the students how to work with algae, what is, because the technology wise each and country which is having some advantages and disadvantages, we will show them how the technology is happening and how we have to take forward new topics to in their future perspectives, future research wise. So we are open to work with different universities also. We will support in the research and we'll also support in the lab activities also. You can come to me anytime, sir.
Okay. Thank you very much, madam. Thank you.
Thank you, Dr. Gupta ji and also Dr. Aman ji for this opportunity to share my knowledge, my experience with all of you. Thank you so much.
Thank you so much, Dr. Ambika ji, respected Director Sir, and dear all students as well as faculty members. Definitely, this will give we can say that helpful in the research area also. So thank you so much.
Thank you. Bye bye.